Uh, Katelyn Conery, are you here?
Yes, I am.
Okay, there you are. You ready?
Picture yourself walking through a mature ponderosa pine stand. The understory is open with tall, sturdy pines nicely spaced around you. As you walk around, you can hear a crunching beneath your feet. All of the senescent pine needles that have dropped from the canopy above are now dry and golden brown. Little do you know, there are thousands of little bacteria and fungi working to decompose all of this pine litter. But do all microbes decompose pine litter the same? Are microbes best suited to decompose the plant material in their environment? Think about it this way. Not all people have the same dietary needs. Some people have food allergies, some people are picky eaters, and some people just eat everything. Could the same be true for different microbial communities? Are some communities sensitive to their nutrient source? Are some picky decomposers? And can some decompose whatever is put in front of them? My research seeks to understand how different microbial communities, each from a different environment, decompose the same type of plant material. In this case, the pine litter from the forest we were just walking through. My preliminary data do suggest that the microbial community composition matters, with mass loss of the ponderosa pine litter varying across these different communities, as depicted here by how much of the leaf is missing. Microbial respiration also varied across these communities, suggesting that they're decomposing this litter differently. Next, we want to connect who is doing the decomposing with these different decomposition rates. So we have sequenced these communities and would like to link certain microbial taxa with these different processes. We want to know who's picky. My research contributes to the unraveling of microbial community dynamics in carbon and nutrient cycling. So as litter is decomposed, compounds are released and cycled among organisms in the system. So understanding these phenomena, the decomposition of organic material, building soil organic carbon, and the use of soil carbon stores that are then respired and released into the atmosphere can be essential for mitigating climate change. Next time you're walking through a ponderosa pine stand, think about all of these little workers right below your feet. They can have a much larger impact than you might think. Thank you.
Thank you. Thank you very much. How did you get interested in these microbial communities?
So I've always had an interest in like forestry and natural resource type sciences. And this project was here and I was able to put my own kind of forestry spin on it, as long as it dealt with some microbial ecology. And so I took it on.
Excellent. So where do you see it going when you're done?
I think there's just a ton of work that still needs to be done with microbial communities. And so this is just a little snippet of really understanding community dynamics and how they're using the inputs from forests essentially.
Super. Well, thank you very much, Katelyn.
Thank you.